So my name is Damien Cabral. I am a fourth-year graduate student here at Brown, working in the lab with Peter Belenke. I've been working on microbiome stuff for just over 2.5 years now. And today I'm going to be talking to you about analysis of 16S data using this relatively new analytical technique called DADA2. So with that, so just a quick overview of what I'm going to be talking today. So I have two goals for this tutorial. The first goal is to be able to implement DADA2 on your own data in R. This is actually going to be probably the easiest part. Most of this code, you can honestly copy it and tweak a couple of things, and you'll be able to run it on your code just fine. But what's more important is I would want you guys to have at least a general understanding of what DADA2 is doing and how it works, and how it differs and is possibly superior from other alternative strategies. So just brief outline, going to go a little bit of brief background on DADA2. I'll go through a complete DADA2 workflow. And at the very end, I'm just going to do a little bit of Phyloseq, but that would be a full Phyloseq tutorial could stand on its own, so we don't have time for that today. So a couple of disclaimers before getting into it. Kellyanne mentioned that Chime2 is now up and running, and Chime2 has really vastly simplified and condensed the DADA2 workflow and integrated it with a lot of other kind of analytical pipelines. And most of the steps that I'm going to show you in this tutorial have been condensed into one or two lines of code. So it's a really powerful tool, but at the same time, it risks becoming kind of a black box. So what I'm hoping to do in my tutorial today is to understand what's going on inside of that black box so you know exactly what's going on with your data and how you can tweak it to suit your particular needs. Additionally, Chime2 is developing a graphical user interface. So if you're kind of new to this field, this is a less daunting version than coding or the command line interface for analyzing microbiome data. So right now, there is a bit of a trade-off. So DADA2 is not quite as customizable if you run it through Chime2 as compared to running it as I will in R. But I'm sure that will change as Chime2 is continually improved and updated. So this graphic summarizes what DADA2 is trying to do. So in an environmental sample, you have, say, 4 unique sequences. During the course of amplification, these errors arise, and they're similar sequences, but slightly different. And that introduces some noise into your data set. So the traditional strategy up until this point has just been to kind of clump everything into these OTUs. Usually it's at a 97% threshold. You can do 99% or 94. But what it does is it just groups these clusters into sequence similarities. And from this figure, you can kind of see the inherent limitation to that. Even though the red and green are different, the OTU clustering in this case is putting them into one OTU, even though they're coming from something different. So what DADA2 tries to do is to resolve some of these errors. And what it's actually doing is it's actually doing sample inference. So based on your sequences and the quality scores and their abundances, it makes a call as to whether or not they're actually real or not. And based on that, it'll resolve some of these errors and get you a more fine-tuned answer. So a couple things about traditional OTU selection. So as I mentioned before, it's slightly low resolution. So a lot of times things are clustered at 97% similarity, but sometimes 97% similarity just isn't enough. So a lot of times it's not enough difference to really differentiate between two species in a genus. Sometimes it's not even good enough to resolve two closely related genera. For example, Klebsiella and Escherichia normally are grouped together because they're so similar. Additionally, for at least the de novo OTU clustering, it's highly dependent on the data that you're actually inputting. So as you get more data from more samples, you have to rerun the clustering based on what you're inputting. So that's very time and work intensive, as you can imagine. Additionally, this strategy can lead to a lot of singleton reads, and a lot of these times singleton reads are most likely caused by just plain sequencing errors. So this will overinflate your OTU count and give you a lot of false positives, so to speak. Yep.
So, I'm sorry. If so, you're saying that there is an possible inflation with the traditional method, like you can't resolve same species within a genus or something. But if the amplicon that I'm choosing to amplify in a 16S case, so whether or not I choose to run DADA2 should be whether the amplicon is supposed to be used beyond the genus level classification or not. Like if the choice of the region I amplify itself limits me in accuracy as to which level I can go down to.
Yeah.
I'm dependent on that. I can choose whether or not I need DADA2.
Yeah, so DADA2, I'll go into this a little bit more. Okay. Since it resolves some of those, in some cases, single nucleotide differences, it's actually better if you want to get down to like the strain level or species level. But depending on the hypervariable region, that just might not be possible. I'll talk specifically about V4. It's very difficult to get down past genus, no matter what program you use. So DADA2 is better if you want to get a specific, fine detail answer, but you're always going to be limited by your hypervariable region, regardless of what strategy you're using. So some benefits to DADA2, it's actually compatible with all amplicon types. So here I'm obviously talking about 16S data, but you could use it on 18S data, 16S, ARPO-B, pretty much anything you want with some just slight modifications to the pipeline. Even though DADA2 was designed to work on Illumina data, you can actually use it on ion torrent and 454 power sequencing as well. Once again, it requires a couple of modifications to this pipeline that I'm not going to go into today, but it's definitely possible to do. As I was explaining to Namrata, it provides single nucleotide resolution of amplicons, and I'll talk a little bit later how that aids in the identification of chimeric sequences. And also, because it takes into account the error rate, it lowers the amount of OTUs and in general lowers your false positive rate. So before we get into analyzing this data, just a couple of quick notes before starting. Before you input your data into DADA2, barcodes or Illumina adapters should be removed. This is the case for our data, so we don't have to worry about that today. But a couple of tools that you can use are cutoffs or Trimmatic, as Kellyanne demonstrated during her tutorial. Another thing is that samples should be demultiplexed into individual FASTQ files. So most sequencing facilities will do this for you if you provide them the barcode sequences. They can split them up for you. But sometimes you might just get a couple of big FASTQ files with a corresponding indexing read file, and you need to split them up. So here are 3 tools that you can use for that. My personal favorite is this first one called idemp. So idemp is really simple to use. You just input a text file with your barcode and the corresponding sample identifier that you want to assign to it, your indexing reads, your forward and reverse reads, and that's it. And it spits out separated FASTQ files for you. Some other tools are FastX Toolkit. I haven't personally used that myself, but I've seen it used commonly. And you can also use Chime1 scripts to demultiplex. The developers don't recommend this because for one reason, Chime1 is being phased out and it will not be supported starting in January of 2018. And also, it's a little bit trickier. You have to pass a couple of extra flags to make sure that your data still stays in a usable format. And lastly, if you're using paired end data, the forward and reverse reads must be in the same order. So this normally isn't a problem. It can be if you're using the Chime1 scripts. But this just allows reads to successfully merge towards the end of the pipeline. So the data that we're going to be using today, they're paired end 250 base pair reads generated on an Illumina MySeq. And they're amplicons of the V4 hypervariable region of the bacterial 16S rRNA gene. And they're amplified using the 515F and 806R primers. So these primers are very standard. They're probably the most commonly used primers to amplify 16S. And they're published through the Earth Microbiome project. So if you want to use these primers, you can go on the Earth Microbiome page website, and they have all of the primers there for you. One thing to note about this, the insert that we're getting from this amplification is about 290 base pairs. So this is a really important thing to note kind of before you start data analysis, because since we have these paired and 250 base pair reads, you're going to get almost complete overlap, which is nice because you can aggressively trim your reads to make sure you don't have any bad quality sequences, but you'll still have enough to successfully merge the reads at the end of the pipeline. Additionally, primers have already been removed from the beginning of reads, so you can do that, as Kellyanne mentioned, in Trim-O-Matic by cutting off the first 18 to 20 base pairs of your reads. If you want to do it inside the framework of this workflow, you can actually modify it to cut off those first reads as well. With that, we are going to begin the DADA2 workflow in the Python notebook. So the way this is going to work is on the slides, I'm going to put up a block of code. That block of code these blocks of code correspond to what you see in your Python notebook, and you're going to run it. And I'm going to have some notes and some things to consider at the bottom, specifically so you can specifically tailor it to your needs. So since we're a little bit short on time, normally I would suggest you run it as I'm going through it, but it might be a good idea just to go through the entire file, run all of the commands in order, and I'll talk through them just so you can, the file can be finished running by the end of this tutorial. Okay, so here I just outlined the general DADA2 workflow. So from raw FASTQ files, you trim and filter the reads inside of the program itself. Using these reads, you can you can estimate the error rate based on the quality scores associated with all of those reads. And then using the error rate estimation, you can perform your sample inference with the core DADA2 algorithm. Once you perform that, you can merge the reads and check and remove chimeras. Once you have a clean sequence table with no chimeric sequences, you can assign taxonomy, construct a phylogenetic tree, at which point you're ready to import it into Phyloseq or other visualization softwares. So some of these blocks of code I'm not even going to spend more than 10 seconds on, but some of the more intensive and important ones I'll take a little bit more time and explain what's exactly going on. So the first thing you do in R is you load your packages. For this tutorial, you need 5 packages. So the first one is the DADA2 package for obvious reasons. So that has all of the scripts needed to execute DADA2. This decipher package performs a sequence alignment. And that sequence alignment is needed to construct a phylogenetic tree, which you use the Fangorn package to do that. And the last 2 packages are just ggplot2 and Phyloseq, and those are just for data visualization and analysis at the very end. Okay, so the first thing you do is you just have to point R in the right direction. So you have to tell them where are your files and that you'll be using for this analysis. So you do that using just by specifying the path for R. And then if you if you type out list files for the path, it spits out all of your files. So this is useful because it just confirms that you're in the right place. And here you can see all of the files that you'll be needing for this: the Python notebook, all of the reads, taxonomy file folder that I pointed out, and the mapping file. Kellyanne touched on this earlier, but once again, the R1, all of the reads that say R1 are the forward reads, and all of the reads that say R2 are the corresponding reverse reads for that sample. So the first thing you do is you just sort the files to make sure they're in the right order, because you want to make sure the forward and reverse reads are actually corresponding to each other. And then you extract the sample name. So this is a pretty standard line of code. So what this is doing is all of the files have a structure where it has sample name underscore number.fastq. So what this is doing is it's pulling out that first part of the sample file. So the reason I'm pointing this out is because this is actually important for downstream analysis, because if these sample names don't match what's in your mapping file, you won't be able to correlate any metadata with your samples. And then this just specifies the full file path to the forward and reverse reads. So the first thing you do with your actual data is you plot the quality scores and determine your trimming cutoffs. So the first block is run on the forward reads, and it should look something like this. So in general, these quality scores are actually quite good. They never actually drop below 30. 30 is kind of an arbitrary cutoff, so 30 is a very like stringent cutoff, so you're only getting very high quality data at that point. And you never see the quality scores dip. So for reference, the green line is the average quality score. So that's the thing you should pay most attention to in these graphs. So, however, at the very end of the read, maybe it gets a little bit noisier. So just to be safe, we will trim the final 10 base pairs of this read just to make sure we only have high quality data. So this is showing the quality scores for the first two files. I've seen from my experience that you don't see much variation from sample to sample. You can see these look pretty much exactly the same. So you don't have to check this for every file, just do it for a couple to make sure you're on the right track. So the reverse reads are a slightly different story. This is totally normal. So this is very common with Illumina sequencing, as Ashok explained earlier. And here we see a more dramatic drop-off of the average quality score earlier on in the read. So they're pretty good, and then they start to drop off around 160 base pairs. So we know that's where the data starts to get a little bit dicey. So that tells us that we should cut at around 160 base pairs, give or take a couple of base pairs. One thing to note here, I mentioned earlier that we'll have almost complete overlap with the V4 region just because of how long we know it is. So this allows us to be aggressive with trimming. So we can cut off pretty much as we need to ensure that we're only getting the highest quality data fed into the algorithms. So this is an example of some really, really nice ideal looking data. So this is actually some of my data, which is still nice, but it's not as ideal as the others. So on the left, you have the forward reads, and on the right, you have the reverse reads. And as you can see, for my forward reads, the quality drops off around an average quality score of 30 at closer to 200 base pairs. And on the reverse reads, it's even earlier, it's about 140 base pairs. So if we were running this on my data, you'd say, okay, I should cut this off, the forward reads off at 200, and the reverse reads off at 140 base pairs. So just an example of how you should tailor it, given on how good your read looks, or run looks. So this here, this just creates a new directory for the filtered files. So this is already created for you, but it takes less than a second. And then this just renames the filtered files that are outputted from this from the next command, and just says F for forward, filtered, R for reverse, filtered. So that's pretty straightforward. So the first big step in the DADA2 workflow is actually quality filtering and trimming. And you do that with this filter and trim command. I'm going to go through each of these components briefly. So the first thing you do is you specify where your forward reads are. So that's where your starting reads are, and then you specify where you want to output your filtered, trimmed forward reads. Same thing for the reverse. You put where your reverse reads currently are, and then you specify where you want to put your reverse reads. So the first thing is this trunk length. As I mentioned before, based on the quality scores of this data, we would want to cut the forward reads at about 240 base pairs and the reverse reads at about 160 base pairs. So this structure of this argument is listed here. So forward read is first, followed by the reverse reads. So some things to consider when setting this parameter on your own data is how long is your amplicon and how much overlap you need. I'll talk about this a little bit more later, but DADA2 by default needs 20 nucleotides of overlap to successfully merge the reads, but you can lower that if need be. If you want to keep that 20 nucleotide overlap, you have to compromise with how much you're trimming in order to make sure your reads successfully merge. With V4, you get complete overlap, so this is almost not inconsequential where you decide to trim it, as long as you don't trim it at like base pair 10 for the reverse reads. But for longer amplicons, you'll need to kind of make that judgment call. So like V4, V5. So previously, before sequencing V4, I did V4, V5, and I would have to reduce the amount that I was trimming from the forward and reverse reads. So max n just specifies the amount of ambiguous base pairs that you can have in your data. So as of now, DADA2 actually can't handle ambiguous base pairs. So by setting this to 0, you get rid of all of your reads that have any ambiguous base pairs whatsoever. So until DADA2 changes that aspect, I would highly suggest not changing that. I've never done it myself, but I can imagine it doesn't result in good things. The next, max EE. So this is the maximum amount of estimated errors that you would expect to see in a read. So I won't go into too much detail, but the authors found that this estimated errors function did a better job at approximating the quality of the read than just the average quality score. So I just listed the equation here if you're interested at all. So reads with more than this number will be discarded. So 2 is a good baseline because you don't want reads with a ton of expected errors in it because it makes the DADA2 algorithm take longer and makes it less accurate. So some things to consider when setting this, how much depth are you sequencing and how good are your quality scores? If your quality scores are really good, you don't have to change it. If anything, you can lower it because you should expect to see less errors in higher quality reads. But if you have poor quality reads and you can't sacrifice as many of them, you can increase this number to make that a little bit more relaxed. So the next one is trunc Q. So this truncates your reads at the first base pair with a quality score equal to or less than 2. So 2 is an awful quality score. You wouldn't want this read in your data set anyway. So for reference, a Q score of 2 means that base has a 63% chance of being incorrect. So this, what this does is it really just gets rid of your worst reads that are probably unusable to begin with. So 2 is a good permissive number. You could increase it, but the nice thing about DADA2 is that you don't have to go crazy trying to get rid of every single sequencing error that you have. Since DADA2 will account for those errors, you can kind of just let your data run through the pipeline and not worry too much about it. So the next one is this RM5X. So I don't think anyone really mentioned this, but typically when you do 16S amplicon sequencing, you have to spike in this DNA from this bacteriophage called 5X, and that adds diversity to the sample. So I won't go into too much detail, but that just prevents the Illumina computer from misreading your clustering. So I would recommend keeping this unless for some reason you know you didn't have this 5X in your data set. Otherwise, leave it as is. And these things just compress the FASTQ files outputted, and the multi-thread just parallelizes this action. So this just reduces the amount of memory that these files take up and speed up analysis. So these are good parameters to keep in place. So now that you have your trimmed and quality controlled reads, you can do the first part of the core DADA2 algorithm, which is the error rate estimation. So this command, this pair of commands, will create an error model that will be used by the DADA2 algorithm downstream. So every batch of sequencing will have a slightly different error rate. You can see that when you looked at my data versus the tutorial data, mine was slightly worse while the tutorial was slightly better. So this is really important to rerun every single time. So what the algorithm does is it has to start from somewhere. So it makes the assumption that you have the maximum possible error rate in your sample, which practically means that it takes your most abundant sequence and it assumes that's the only sequence that's actually true and all of the other sequences are caused by errors. For your sake, I hope that's not the case, but it just has to have a starting point. And what this program does is it alternates error rate estimation and inferring the sample composition until it arrives at a solution that makes sense for both of those parameters. So it kind of goes back and forth. So that was done on the forward reads. If you do it on the reverse reads, it works exactly the same way. One small thing I'll point out is it actually takes more of these steps, and that's because the reverse reads are worse. So it has to do more of those cycles to come upon a solution that makes sense. You can then, as a kind of sanity check, plot these error rates to see what they look like. So what this does is it plots the frequency of each possible base transition. So for example, the frequency that an A is actually read as a C, and it plots it as a function of the quality score, the consensus quality score on the bottom. So the black line is what you're actually seeing in your data, and the red line is what you would expect to see in your data under the definition of the Q-score. All I really want to point out here is that as the quality score increases, these expected error frequencies decrease. So this is just like a good check to do before moving forward. So that makes sense because the higher quality sequencing you have, the less errors that you would expect to be in your data set. So when you compare that to my data, some of the minor trends are a little bit different, but for most part, the major theme is the same. So as quality score increases, the expected error rate decreases. All right, so now that you've estimated your error model from your reads, the next thing you do is dereplicate them. So this is just so you don't have, the DADA2 algorithm doesn't have to work on every single read you have, because normally you have hundreds of thousands, if not in the millions of reads. So this just kind of speeds up and simplifies the computation. Okay, so the next part is actually the heart of the DADA2 program, and that's sample inference. So what you do is you feed the error model that you developed a couple steps previous, along with your dereplicated reads. And what this particular command actually does is it calculates an abundance p-value for each of your unique reads. So basically what it does is it's testing the, quote, null hypothesis that a sequence is too abundant in your sample to be explained solely by errors in your data set. So to conceptualize it, a low p-value means that this is actually a real sequence and it likely wasn't caused by just random errors during sequencing, while a high p-value means that it was likely caused by errors. In this case, it would not be that particular read would not be kept. It would just kind of be thrown out because it's low quality data. So DADA2 does that on all of your samples. Does the same thing on the reverse reads. Does nothing really special about that. Okay. So now that you've successfully denoised all of your forward and reverse reads, you can actually merge all of them. So this is slightly different than the workflow Kellyanne presented, where she merged very early on, and then she used that to do her closed reference OTU picking. So this comes after you've taken all of the possible errors into the account. So what you do is you, to this merge pairs command, you supply the data sequences for both the forward and reverse reads, and the dereplicated forward and reverse reads. And the output looks something like that. So a couple of important notes about this. So DADA2, by default, will only merge reads if they exactly overlap. And the reason behind this is because in the previous steps, you should have already accounted for those errors and taken them out of your sequencing run. So if something's not matching for whatever reason, there's a chance that one of the DADA2 algorithm could have made an error. So it doesn't keep those two merged reads. So this is a bit stringent, as you can imagine. So you can change this by adding the max mismatch option to the command above. So by default, it's 0, but you can make it, say, 1 or 2 if you're finding that you're losing a lot, or a lot of your forward and reverse reads are not merging. I mentioned this earlier, but by default, the program requires 20 nucleotides of overlap. That can be a little bit much, particularly for these longer hypervariable regions. So you can lower it to lower it if need be, or alternatively, you can just concatenate your two reads, which just literally sticks them together. Though that can mess up with taxonomic assignment and other things later on. Since our reads almost completely overlap, Don't really have to worry about the setting, but if you are using a region like V4, V5, it's something you should definitely consider, maybe either relaxing the amount of overlap you need or the number of mismatches that you're allowing. And you might have to compromise how much you trim earlier on. So now that you have your merged forward and reverse reads, you can create a sequence table with this make sequence table command. And you can see the breakdown of the size of these amplicons. So on the top row here is the size of the merged read, and on the bottom is the frequency. So for example, 182 sequences are 253 base pairs in length. So this output here, the sequence table, is the most analogous thing there is to an OTU table in DADA2. It's a technical thing, but they don't like to call it an OTU table. They prefer to call it a sequence table or a table of sequence variants, something along those lines. But you can essentially treat it the same way as you do a regular OTU table. So the next step that's really important in this workflow is the Chimera checking and removal. So I don't think we went through what a Chimera is, but it's basically a PCR artifact. So if you are amplifying a region of your 16S gene, your primer will start amplifying the region, but at some point it'll fall off, but it won't go all the way through. So that oligo can then be used to prime another completely unrelated 16S region. So you'll kind of get these amplicons that are actually fusions of two and sometimes more parent sequences. So what this command does is it takes your sequence table and it performs this multiple sequence alignment. So it starts basically from your least abundant read, and for all of the reads that are more abundant, it'll do sequence alignments with all possible combinations to see if there's any overlap. So to illustrate this, you have two parents, a blue and a green one here, and during the course of amplification, you create this child that's half blue, half green. So you align this child read with all possible parent reads. So in this case, you align the child with this purple, dark blue read, and it doesn't align, but it does still align with the end of this green parent. But because it's not completely covering the read, it's not considered a chimera. However, when you perform this alignment with the light blue parent and the green parent, you can, it's very obvious that this child is just a chimera of these two. So the DADA2 algorithm will recognize this and filter it and remove it from your database. From your sequence table. So here, if you run this command here, you just get the proportion of reads that are not chimeric in your data set as a function of how many are. So here, you can see 96% of your reads are not chimeric. So that's a really, really good data set, which shouldn't be surprising because we saw that the data was high quality earlier on. So an example from my data, it's not quite the same. So one thing I want to point out is that the number of chimeras identified actually are a vast majority of the total unique sequences. So 97% of the unique sequences identified by this, 97% of the unique sequences identified by DADA2 are actually chimeric. The flip side of that is that these, even though it makes up a majority of your unique reads, it should not make up a large proportion of your total reads. So these are probably very low abundance in your data set, so maybe like 2 or 3 copies each. But when you run the analogous command, you see that only 71% of my reads are not considered to be chimeric. So that just gives an example. So this is probably one of the more extreme cases, but this just gives an example of how important that chimera checking is in the DADA2 pipeline. So this just kind of summarizes what I read. So if you did find that a majority of your total reads are chimeric, you most likely have some problem. The most likely problem is that you didn't trim off your primers at the beginning of the reads. So because these primers typically have ambiguous base pairs, they can map to several different parents. So it can artificially inflate the number of chimeric sequences you're detecting. So that's why it's very important at the beginning, either before you begin this pipeline or during the pipeline, to trim off the first 20 or so base pairs of your read. So if this were to happen to you, you would just go back to the filter and trim command and use this trim left argument for however long your primer region is. So if that doesn't fix it, there just might be other factors at play. So you could try trimming more low quality base pairs if that's something that you can do with your data set. Another thing is that you might want to think about the hypervariable region you're sequencing. So I'll use V4, V5 as an example. So between the V4 and the V5 hypervariable region, there's actually a constant region between that. So that constant region is a prime target for chimera formation. I think studies have been done where chimera formation is much, much lower if you sequence one single hypervariable region like V4 compared to V4, V5, or V1, V2. So that might be something that if you have a high amount of chimeras and you can't seem to figure it out, that might be something to try it out. Another thing is just how complex your community is. This one's kind of a bummer because you can't really change how complex your community is. My understanding is that communities that are very similar to each other, at least in sequence similarity, are more prone to chimera formation, but there's not much you can do about it. So this might turn out to be something you just kind of have to live with. If that is the case, you can maybe sequence in more depth so that after all of this trimming, you still have a usable amount of reads to do your data analysis. Okay, so at that point, you have a sequence table with clean reads, and you can now assign taxonomy. So you pass it to this assign taxonomy command, the chimera removed sequence table, and here you specify the file path to the particular database that you want to use. So what this command does is it uses an RDP naive Bayesian classifier algorithm to assign taxonomy. So even though it has RDP in the name, you don't have to use the RDP database. That's my personal preference, just because RDP is pretty well curated and pretty well updated. But DADA2 provides specially formatted databases for RDP, green genes, and Silva. So you can use your database of choice. So one weird quirk that I want to point out, I don't exactly know why this is done, but that last command only assigns taxonomy from the phylum to the genus level. My speculation is it just splits up the program and makes it a little bit faster, but I can't say for sure. So if you do care about species level assignment, you run this add species command. So you use the taxonomy table that you just generated, and for at least RDP, there's a separate database that has these species level assignments. So here you just direct it to that. In this command, you're just labeling the taxonomic level, so everywhere from kingdom to species. So one thing that you'll notice is that only 7 out of 215 sequences were identified down to the species level. So that's a really low number, and that's actually what I've seen in my own data. So some possible explanations. I mentioned this earlier, but the V4 region is fairly small, so it might be hard to get a definitive species level assignment with the V4 region. Additionally, my understanding is that the V4 region isn't the most hypervariable of the hypervariable regions. I know for a fact that V1 is a little bit more variable, so if you do care about species level assignment or even strain level assignment, you might want to consider a region that's a bit more variable. Okay, so at this point, you have a sequence table and you have a taxonomy table that assigns taxonomic classifications to each of those sequences, but you don't have a phylogenetic tree. So if you want to do anything that involves phylogeny, you need to make one. So what this first command does is it just aligns all of the sequences in your sequence table. And what you then do is you pass that sequence alignment to the Fangorn package, which then can construct a phylogenetic tree. So it starts off using neighbor joining method. and then it subsequently uses that tree to create a maximum likelihood tree from your data. So this is one that I haven't really played around with too much, but I know there's other programs like FastTree that can also generate trees from your data. Okay, so that should give you a phylogenetic tree. So now you have tree, sequence table, and taxonomy table. So now you're ready to actually analyze and visualize your data. And I'm going to go through this last part really quickly, but the way I'm going to show you how is in Phyloseq. So what you do with this first command is you import your mapping file, which usually looks something like this. So this can be have any column you want. So the only thing that is important is that the first row of the first column has this hashtag sample ID identifier. But any of these other categories are up to you. You can fit to be your your particular data set. So what this this Phyloseq command here does is it takes all of the components that we have, so taxonomic table, sequence table, tree, and mapping file, and it kind of puts them into a usable format. So it'll associate the metadata that we showed in the mapping file with all of your samples in your data set. And this will allow you to later analyze and visualize your data. So I'm not going to talk too much about the experiment here, but the metadata from this particular experiment, it's just the gut microbiomes of mice at various points post-weaning. So they're all female mice. This just shows the day at which that mouse was sacked. And then this is just a categorical variable that they added, so splitting things up into early or late mice. So one other tidbit before moving forward. So the phylogenetic tree that we made currently doesn't have a root. So if for some reason you don't care about phylogenetic relationships, this isn't that important. But otherwise, you absolutely need to do this before moving forward. For example, I've forgotten to do this in the past, and when I go to calculate like unifrac or make a PCOA, I'll run the command several times on the same data set, and it will change. And I'll get a different answer almost every single time. And that's because what the program does is it randomly assigns a root to your data. So if it just happens to assign a completely different root, you could get a very different answer. So we won't really do anything with phylogeny in the next part, but this is something that you should definitely do if you want to calculate like unifrac, either unweighted or weighted, or like Faith's phylogenetic diversity. Other metrics like Shannon and Simpson diversity and Bray-Curtis dissimilarity for beta diversity don't need phylogeny to be calculated. So the first thing we're going to do is just plot the alpha diversity metric. So I'm going to, so to this command, you feed the Phyloseq object that you just created. On the x-axis will be the day category of your mapping file. And what this is showing here is that it's going to calculate both Shannon and Simpson diversity measures for your data. This command here just will color your points based on that categorical when variable, so early or late. So if you run the command, you'll get this error message. And I just want to take an aside to explain this error message. So it says that we've trimmed the data and that there's probably an issue with it. And you'll notice that we actually didn't do that at all in our data set. And that's because DADA2 does not allow singletons to pass. So as I mentioned earlier, DADA2 calculates a p-value for each sequence that you provide it. And singletons, by design, always have a p-value of 1. So it will never be classified as a true unique sequence. And this is done by design by the DADA2 programmers because it's really, really difficult to differentiate true singletons from sequencing errors, even after the DADA2 algorithm. And if you include them, they can artificially inflate your diversity. So they just kind of do away with them completely. So this command will generate a plot for those alpha diversity metrics, but if you just want the raw numbers, you just run this estimate richness command, and it'll give you those values, and you can plot them any way you want. So when you plot it, it should look like this, with Shannon diversity on the left and Simpson diversity on the right. Don't have time to go into too much about Shannon and Simpson diversity, but Shannon diversity typically is more a measure of richness, and Simpson is more a measure of evenness of the community. So both follow the same general patterns here. That's not always the case because they're calculated in very different fashions, but it's just nice to plot or have a couple of different diversity metrics when analyzing your data. One other thing I'll mention, so obviously DADA2 gets rid of singletons, so that means some diversity metrics are completely inappropriate for DADA2. So for example, chow1 is calculated based on the abundance of singletons and doubletons. So you can see the problem. If there's no singletons in your sample, you can't really calculate chow1. But Shannon and Simpson diversity work well with this particular metric. So last thing I'm going to mention is beta diversity. So Kellyanne showed beta diversity using weighted and unweighted unifrac. Using a PCOA. So I show here actually a different method of ordination. So here it's a NMDS, or non-metric dimensional scaling. I won't go into the ins and outs of how it's different from PCOA. But what you do is you can ordinate your data using this NMDS method, and you can specify the distance metric. So Bray-Curtis dissimilarity doesn't take into account phylogeny. It's just a little bit different than unifrac. So when you plot that, you can see your samples kind of clustered together in two different batches. So the early samples clustered together and the late samples clustered together. So you can use the same commands to plot PCOA just by changing this NMDS to PCOA. So that's all I have for the tutorial. I know that was a lot thrown at you in a very short period of time, but for more information, I can direct you to the original paper for DADA2. So if you care about how DADA2 sample inference actually happens, it's a great place to read about it. Additionally, you can go on the GitHub page for DADA2. A lot of that tutorial was drawn from that. And additionally, they published an F1000 paper in the past couple of months. So that's where I got a lot of my ideas for the phylogenetic tree generation. And with that, that's all I have. So thank you. Yeah.